So in this brief presentation, I am going to explain about the types of point mutations. Point mutations in fact refers to nucleotide changes in the DNA molecule. We know that ATGC bases are arranged in a specific sequence in the DNA molecule, and any change in such sequence is referred as point mutations. We use a number of terms to explain the specific nature of point mutation. So all such aspects I am going to present in this presentation. See, the point mutations occur randomly, that is without regard for any possible benefit or disadvantage to the organisms. But definitely those mutations which occur in the protein coding genes, and if they show benefit to the organisms, they are considered, they are actually selected, so they come under selection pressure, and because of such reason they get propagated in the population. And mutation alone creates new alleles in a gene pool. This is another important thing, that variations among the different individuals results due to mutational changes, because it is mutation that actually creates variation in the genetic content. So mutation alone creates new alleles in a gene pool is an important thing to consider. And there are two terms like transitions and transversions, which are quite often used by the geneticist. Let us get acquainted the meaning of these two terms. See, A and G are two purine bases, and T and C are two pyrimidine bases. So adenine being a purine base, if it changes into another purine base like guanine, then it will be transition. And it could be vice versa, that is a guanine may change into adenine. So here this arrow is on both sides. It is indicated that A may change into G or G may change into A. So this kind of change will be transition. In the same way, if a pyrimidine base like cytosine changes into another pyrimidine base, that is thymine, or just thymine changes into cytosine, then this kind of change or substitution will be referred as transition. So change of a purine into another purine or pyrimidine into another pyrimidine will be referred as transition. So this way 4 different types of transitions are possible. Okay, it has become clear that if adenine is there in the DNA and if that is replaced by G, then it is a transition mutation. Likewise, if there is C in the DNA sequence and if it is being substituted by T, then that will be a transition mutation. But transversions are different mutations. In such cases, a purine base may change into a pyrimidine base or vice versa, a pyrimidine into a purine. So this will be referred as transversion. See here, if A being a purine base changes into T, that is a pyrimidine base, then this will be transversion. Or this T changes into A, this will be a transversion. So here also arrows are indicated in both ways. So if you count the number of transversions, then it would be 2 to 4 to 6 and to 8. So 8 total transversions could be possible. In you can say that total 12 kind of substitutions may be possible in the DNA molecule. Then we use the term silent mutation or missense mutation or nonsense mutation or phrase mutation. These are 4 terms which are used to explain the type of mutation. So let us see the meaning of silent mutation. But before I explain this aspect, let us get acquainted with the DNA sequence. Suppose in the DNA sequence, the sequence is T, actually DNA is double stranded structure, but I am showing the template strand, which is starting from a 3' end. So it is TAC, AAA, AAA, GAG and ATC. Suppose a small segment of DNA is being shown, and that too single stranded DNA, and this one is a template one. From this DNA template strand, the mRNA is synthesized. So this mRNA is being represented as 5' and AUG, because T will encode its complementary base will be A. Then in messenger RNA it will be U coming at the place of A, because T will not be there. So C, its complementary base will be G. So this will be the messenger RNA bases. These bases form the messenger RNA, like AUG, UUU, UUU, and then CUC and UAG. So you can exactly know that which base will be coming in the messenger RNA by observing the kind of base present in the DNA molecule. And then this messenger RNA will be used for protein synthesis, and we know that 3 later make a codon. So AUG, being initiator codon, will be recognized for methionine. The next UUU, it will encode for phenylalanine amino acid. Then this will be phenylalanine. CUC encodes for leucine, and UAG, being a terminator codon or a stop codon, here the protein synthesis will stop. So in the polypeptide chain, you can say 1, 2, 3, 4 amino acids will be there. So this is the way the you can say transcription takes place and the translation occurs in the cellular system. Now what will happen if mutation occurs in this DNA molecule, particularly in the template DNA? If mutation occurs at a point, then what would be the situation that we can understand? Now this is genetic codon. Just at a glance we can have idea about it. We know that total 64 codons are possible, and out of these 64 codons, 61 encode for specific amino acids, that is 20 amino acids are encoded by these 61 codons, and 3 codons are stop codons. They are UAA, UAG and UGA. These are 3 stop codons. The initiator codon is AUG. And another thing you can notice here, that a single amino acid, a single like phenylalanine, may be recognized by 2 codons, UUU and UUC. Leucine is being identified by 6 codons. So a single amino acid may be recognized by more than 1 codon, except for tryptophan, which encodes for which is identified by UGG. The most of them they are having more than 1 codon. So here we can have the idea about silent mutation. See, in this DNA molecule, we have just seen the way nucleotides are arranged. Now we have to see if mutation occurs in this DNA molecule, then what happens. See, suppose mutation occurs at this point. See, this AAA, the third letter of it, that is changed by G, means there is a substitution mutation. Now this A is being replaced by G. So it's a kind of transition mutation. And due to this transition mutation, what will happen? After transcription in the mRNA, the codon will be AUG, UUU and UUC. Instead of UUU, it will be UUC. And then rest of the things will be as such. So what will happen? This UUC will encode for phenylalanine. UUU that also encodes for phenylalanine. So we are observing that there is mutation in the DNA molecule, because this is the DNA molecule in which mutation has occurred. For this purpose, I have written here mutation, means it's the same DNA, but it is experiencing mutation at some point. But because of this mutation, change in the codon has occurred, but this change has not resulted into change in the amino acid sequence in the polypeptide chain. And this kind of mutation will be referred as silent mutation. Means mutation has occurred, but it did not affect the amino acid sequence in the polypeptide chain. Next condition could be missense mutation. It means that mutation results into change in amino acid sequence. See here, this is the template DNA. Now this is mutated one, and in case of mutation, at this third letter, means AAA, this third A, it is being replaced by C. Okay, side to C. So as a result of this, in the messenger RNA, the codon will be UUG. Instead of UUU, it will become UUG. And this UUG will encode for leucine. So here, this phenylalanine is now replaced by leucine amino acid. So in the polypeptide chain, we find that there has become a change in the amino acid sequence, and this will be referred as missense mutation. So missense mutation, that always indicates for change in the amino acid in the polypeptide chain of the protein. Next situation could be nonsense mutation. So in case of nonsense mutation, what happens, that mutation results into termination of the protein synthesis. So we can understand this, that this is normal DNA sequence, and now mutation has occurred in it, and this mutation has occurred, suppose in this layer, in this portion, that is AAA, and here two mutations have taken place. This A is being replaced by T, and the another A is replaced by C. So when messenger RNA will be synthesized, it will have UAG, and this UAG does not recognize for any amino acid. It is actually stop codon, so protein synthesis will get terminated over here, and a complete polypeptide chain will not be synthesized. So this is referred as nonsense mutation. And the fourth one is frame shift mutation. Frame shift means the entire frame of the messenger RNA of this codon system that gets affected, shifted due to deletion or addition of nucleotide in the DNA. So one thing one should understand forever, that frame shift mutation occurs either due to addition of some nucleotide in the DNA or due to deletion of nucleotide in the DNA. So see here what kind of mutation has occurred. TSC is as such, then AAA it is as such. AAA, here 1A is deleted. I am showing here a deletion mutation. So A is deleted from this position. So what will happen? AAA, and then it will be G coming over here, then AGA will be coming over here, and TC will be left. And we know that during synthesis of protein, the codons are recognized in continuation. So 3 letter will make codon system, and when the protein will be synthesized, then any change may happen, means that may result into a nonsense mutation, or that may result into missense mutation. Very rarely silent mutation would be taking place, because frame shift mutation may result into severe effect. And here in this case, this UUC encodes for phenylalanine, but next one, instead of leucine, it is serine coming over here, and then you are not getting any amino acid over here, or termination is not occurring. So anything may happen due to this frame shift mutation, and that you cannot exactly deduce until and unless you analyze the entire sequence pattern in the DNA. Now, other two terms which are used are synonymous mutations and non-synonymous mutation. Let us see what does this synonymous mutation mean. If amino acid in the polypeptide chain is not changed due to mutation, then that is referred as synonymous mutation. Means silent mutations, they cause synonymous mutations. That is, mutation has occurred in the DNA, but that has not influenced the amino acid sequence in the polypeptide chain. But if a mutation results into the change of amino acid in the polypeptide chain, then that is referred as non-synonymous mutation. So missense mutations are non-synonymous mutations. The other term which is also in use is neutral mutation. See, neutral mutation is actually mutation that does not influence the fitness of the individuals. That is, mutation has occurred. That could be silent mutation or missense mutation, but that is not going to affect the fitness of the individual. So such mutations are referred as neutral mutations, and neutral mutations may be synonymous mutations, or such mutations may be non-synonymous mutations. So I have narrated some of the salient points regarding point mutations, and which means all these things are very basic informations, but such basic informations are very genuinely required to be known. So I think that this much information would be quite useful to the students.